So I'm going to start off by just making clear what we're talking about today. And I think that's the basis for a lot of confusion when people talk about genetics. So genetics is the science of heredity, or it's the transmission of genetic information from one generation to the next. And I remind people, and this is important, you get the genetics from your parents, okay? You don't give... Somebody once told me that their kid had made them crazy and that it was genetics. And I said, that's just environment, that's not genetics. It's transmitted from parents to offspring. Genomics is the discovery of new genes. It's gene information, it's gene mapping, it's DNA. It's all that molecular stuff that goes along with the genes that are underlying the genetics of the problems we talk about. So, anybody who's taken a genetics course always remembers this very simple equation. Genotype plus environment equals phenotype. And we all know that there's different genetics and there's different environments that pigs are raised. And some phenotypes look like this, and some look like this, the Chinese pig that we helped to bring to Iowa State and to the U.S. several years ago. So we need to keep this in mind. And our role as geneticists, or genomics people now, are to understand those different individual genes and how they're involved. And a few years ago, National Geographic had a really interesting section on secrets of the gene. And my comment is that we can now employ modern methods to understand genes, to unlock the secrets of the genomes of animals. And I'm not just talking about pigs, but I'll make a few comments later on about how this affects humans, because we talk now about people, and we talk about genomic medicine. You may have heard that. If you read Time Magazine or Newsweek, you would have heard the term genomic medicine. And what that means is, again, understanding people's individual genes, and in composite, their genomics, to predict what kind of treatments they should have, what kind of diseases they're likely to get, and so on and so forth. So central to this, and this will be the only underlying biology slide I really have today, is that I'll remind you that animals have cells, cells have chromosomes, those are made up of DNA, the genetic material, and that genetic material codes for proteins. Now, this is what most of us learned in biology. We now know it's considerably more complicated. There's a lot of other things going on. But for the basis, the simple rules of the game, this is it. This covers everything that I'm going to talk about today. So, what about the tools that we have as breeders? So I don't know everybody in the room. I obviously see some faces I know. And some of you work for breeding companies, and some of you are seed stock breeders, and some of you are commercial producers. So DNA markers are groups of bases of DNA that have unique physical locations in the genome of an animal. And their patterns vary. But we can use those patterns to predict the outcome of certain traits. So in other words, a genetic marker may exist in some animals and not in others. And we can use those genetic markers to predict how well animals will perform. And the most common marker, and the one you'll hear me talking about today, is what's called a SNP, SNP marker, or single nucleotide polymorphism. Now, yeah, that sounds complicated, but it really isn't. All this means is there's a mutation somewhere in the genetic code of an animal, and that mutation is associated with a difference in the protein that's produced, or a difference in the expression of how that gene acts. And what I'm doing is I'm just trying to find those markers. And this is akin to, very simply, driving up Highway 35 and looking at the mileage markers. And I know when I get to mileage marker 111, I got to turn if I'm going to get to Ames. And if I'm going to Ankeny, I get off on mileage marker 90. These are, if you like, mileage markers along the DNA of a chromosome in animals. And they vary both within breeds and across breeds. So, as geneticists, there's all kinds of ways we can find these effects, these mutations, these SNPs, if you like. And some are called major genes. I'll give a great example of that. candidate genes, so we do what's called scans or expression studies. We do associations, and all of these methods require validation. And I'm going to present some of the results today and then tell you how they're useful. So I want to start off with the halothane locus. Our first speaker was the Loren Christian speaker today. The halothane locus was discovered by Loren Christian. And he did it before we even talked about DNA. He did it because he used a test. He used some great biology to discover that muscular pigs were dying, and that muscular defect looked to be recessive. And then that phenotype was associated with porcine stress syndrome, and in the early 1970s he realized it was recessive, and he could detect it by using halothane gas because he realized it was very similar to a human condition, malignant hyperthermia. And here's a pig. This is actually an old photograph from a slide that was Loren's. Here's a pig dying from porcine stress syndrome. Here, Loren is in the right there, the guy with the slightly balded head. Screening pigs with halothane. When I first came to Iowa State, once a week we'd go out to the farm. John probably remembers this. You'd go out to the farm and you'd screen pigs. And if you were a novice like me, you weren't allowed to put the gas on the pig, because a pig that had the defect, if it had got too much gas, would drop dead. A pig that didn't have the defect, or was a heterozygous animal, he'd sleep. So you had to be keenly aware of what that pig was doing. He was either dropping dead or sleeping. And you had to be real careful about that. And this was a great test, but it had two drawbacks. Not everybody has a halothane machine on their farm. And secondly, it only found those animals that were recessive, none of the carriers. So what we really needed was a DNA test to do that. And this just shows what happened with hal gene effects on quality. The normal ham is in the center, dark, firm, and dry is on your left, and pale, soft, exudative is on your right. So Loren and some others using blood markers mapped this to chromosome 6. Later, a DNA test was discovered in Canada. It was patented, made available, and the test was used. This was the first genetic test for a simple recessive in pigs, and was used worldwide, and actually stress numbers were reduced greatly. Now what's interesting is there must have been a few companies at the beginning that didn't do a very good job testing, because the frequency of the allele, the bad allele, still exists in rather surprising numbers in some populations. The other approach is to do what's called candidate genes. And here's a cover from Scientific American which asks the simple question, are athletes born or are they made? And the question comes from that because are there biological differences based on our genetics? And we all know that. We only have to look around the room. Some of us are bald. Some have great heads of hair. There are genetic differences amongst all of us. So we use this in our lab, and we've used this rather successfully to look at a number of traits. And one of the ones is, here's a picture of a Meishan pig with 40-odd piglets, and Egbert, she kept all those pigs, so she must have known what she was doing. Chinese pigs are well known. The real issue in most Chinese pigs' litters, and which fits very well with the results from the first test, is all those pigs are born small, which runs against what you think initially, but they're for the most part uniform. And that makes a big difference. Well, we asked the question, why so many piglets? And we started looking at a gene called the estrogen receptor. And estrogen, as all the women in the audience know, is the primary female hormone for nearly all the primary reproductive functions. Well, the receptor is like a glove, and it catches the hormone, and if the glove isn't just right, has a mutation or something happens and it changes things. We found a mutation in this gene, and that mutation is in fact associated with litter size. And let me see if I can get this to work. But if you look here, the AA genotype, which is the least prolific, had 11 born, 11.2, and the BB genotype, 13.5. So about a 2.3 pig difference. These were pigs that were half Meishan, half large white, or half Yorkshire, however you look at that. So a large difference, 2.3 piglets in half Meishan pigs. Now we don't really want Meishan pigs. They're fat, they grow slow, they weren't productive except for litter size. But if you look in U.S. pigs, large white or Yorkshire, or you look all over the world at those two breeds which have this B allele, because this B SNP, if you like, doesn't exist in all breeds, the difference was about 0.8 of a pig, a little over 0.8 of a pig. But that effect, which I call the ESR effect, is 5 times greater than the normal response to selection. So if I can identify genetic markers that are associated with big effects, I can make considerable differences on top of the selection progress that I can make. And this was the first big gene test available on a quantitative trait, not something that was a simple recessive. Now another one I want to show you today is one that's also quite interesting. And this again is a candidate gene. And this was a candidate gene that we discovered from looking at data in other species, mice of all, which we were looking at. Not us personally, but we read in the literature. Here are 2 mice. If you look on your left side of your screen, they're full sibs. And what do you notice? Well, you either come to the conclusion that someone stepped on this poor guy and he just got flatter, or he's just much fatter. And in fact, in mice, as in humans, there are mutations in this gene, MC4R. It's a gene that's a receptor in the brain, and the gene says eat, eat, eat. And if you're unfortunate, in humans, there's about 25 mutations. If you're unfortunate enough to have one of these poor mutations, you will be grossly obese no matter how good you are at the table. It's very sad. And there are people that they talk about genetics, they can't do anything about it. And that was the same with this mouse. It has a gross mutation that forced it to eat and eat. Now, we don't want gross mutations in pigs. We may be looking for subtle mutations that would affect the growth rate, the back fat, and the feed efficiency. And my student, Kwanso Kim, found that, very cleverly found that mutation. And what that mutation does is you can choose one of 2 genotypes. If you choose the growth genotype, the pig eats more, grows faster, and grows a little fatter. If you choose the one that's the efficient genotype, the other SNP, if you like, it grows slower, it eats less, but it's more efficiently converts its feed to growth. And what's happened in the industry is 5 years ago when this first, or 10 years ago when this was first discovered, people chose the growth allele. They wanted faster growth. Corn was cheap, buildings were relatively cheap, facilities, and so what happened? They chose the growth allele. Now corn's gotten very expensive, and some of the swine breeders are fixing the lean allele, making the pigs more efficient. The other interesting thing is that if you choose the efficient genotype, the pigs will produce, they're more efficient, and they'll produce less manure, and you have less environmental issues if getting rid of manure is a problem for you. Now the third way to look for genes is something called a scan. And this is just basically crossing animals from 2 different types. I see Kelly here in the crowd. He's a big Berkshire breeder. We started out looking for meat quality genes, and we worked with Berkshires and Yorkshires. Obviously, one of those breeds had much better meat quality than the other. And then we follow the genetic markers through the population along with the different traits that we measured. And if you do that, you can create a map, and on this genetic map, here's the 18 chromosomes plus the sex chromosome. You can map regions in the pig genome that are associated with different performance traits, different meat quality traits. And as it turns out, we found 2 genes in particular, one that's in very high frequency, the preferred alleles in very high frequency in Berkshires, and that's this gene PRKA gamma. And CAST, another one, associated. And both of these are associated with appearance, color, tenderness, aroma, juiciness, and flavor. So here's a couple of genes that we found associated with improved meat quality. So where are we today? Well, there's a number of genes, this laundry list on the left here. The genes are in 2 colors, blue and black. The ones that are in blue are genetic tests that are available in the industry. So I don't know how many people are seed stock breeders, I don't know how many people work for companies, but these tests are available. The ones that are in blue are available for any company, any seed stock breeder. And in fact, I left a sheet on the 2 chairs that are in the corners. You can pick that up, and this is the genotyping company in Omaha, Nebraska, GeneSeek, that does some of the genetic testing, and the tests are available through them at a pretty reasonable price. There are a number of tests listed here that I list as private. And those are tests that different breeding companies have developed, or different breeding companies have unique IP on, or they have exclusivity of the patents that exist. But all the ones in blue, and many of these are from Iowa State, are now available to any breeder. And I would encourage breeders to consider them. Certainly, if your interest is improving growth rate or feed efficiency, MC4R. If you have large white pigs and you want to improve litter size, ESR and EPOR. And if you want to improve meat quality, you're selling primarily in the meat quality, PRKA gamma 3. Now, this is an interesting one. I'll just make an aside here. The Berkshires have nearly fixed the beneficial allele. So you'd say, well, great, they don't need to do anything. But actually, there's about the 15% of the Berkshire pigs that have the bad allele. That allele could be easily removed from the population. Their mean meat quality improved considerably in the breed, done pretty easily with just testing the sires in the breed. So how does this happen? Well, you can use genetic markers by marker-assisted introgression. That means breeding from one breed into another and carrying the genetic marker along. You can do marker-assisted selection, in which you use breeding values like Egbert talked about today, and you include a few genetic markers. Or you can replace all that you can replace phenotypic selection with what's called genotypic selection. And I'll comment on that. What you get by using markers in marker-assisted selection is improvements for average daily gain and fertility up to about 40%. For carcass traits, which of course we can't measure on the breeding animals. If we did, we'd have to kill them, and then they wouldn't be of much use to us. But on carcass traits, we can make enormous improvements up to 64%. So let's take a look at that. And I'm going to give a couple examples here. So if I'm a breeder, and for a number of years breeding companies had way too many individuals that they couldn't select all the individuals to go into their testing facilities. Let's say they were testing for growth rate, they had fire feeders for feed efficiency, they had maybe too many males in each litter, too many females. So which ones to put into the testing program? Well, the problem is it's a guess. But if you look at it differently, and you actually have a genetic test, you could choose those that have the preferred SNP, the preferred genetic marker, and only put those with the preferred marker into the testing program. So you got the best ones based on first stage selection, and then second stage selection is based on the testing program, and you can celebrate with a good bottle of bubbly. Another place that DNA can be used, and we hear a lot about this now, every time some problem arises with some disease on the farm, is to use DNA for traceability results. And not very many companies are doing that, though there's a lot of interest in some of the big supermarket chains. So identity preservation, from, as they say, farm to fork, DNA is the only way to do this completely. And you can do this, you can look at the different systems that are out there, but DNA has the best. And you can also go one step further, and you could certify products based on DNA. So we could come up with, for instance, a set of markers that are predominantly Berkshire markers, or predominantly Duroc markers. So when I sell pork and I say, this is Duroc sired pork, I can stand behind that from a DNA standpoint and know what I have. So what's going on in the industry? Well, breeding companies, depending on their size, their experience, their level of incorporation of some of the science, are using anywhere from 2 genetic markers to hundreds. In fact, I heard a talk last week or 2 weeks ago from one of the larger breeding companies. They're using panels of between 300 and 500 markers to make selections for each of the traits. So the bigger companies are using hundreds of markers. The smaller companies are using fewer. For all seed stock breeders, there are tests that are available, ESR and EPOR for reproduction, MC4R, CCK, HDMA for growth, and then, as I said earlier, PRK, gamma, and CAST for meat quality. And these are available and can be used, and myself and my colleagues at Iowa State are more than happy to help breeding companies in using these properly to make genetic improvement. I said there's a handout in the back. This is an old handout, what it looked like. But testing's done in North America by GeneSeek in the U.S. and DNA Landmarks in Canada. So just for a moment, let's kind of summarize what I've talked about. 31 years ago when I started, when we wanted to make changes, we used different terminal sire lines. So line choice was the beginning point. So I would go to a breeder who I trusted, or a breeding company. I would choose a certain line of boars, or a breed of boars, however you want to look at that, and I would use that to make certain improvements in growth rate, meat quality, and so on. Later, as we got best linear unbiased prediction, or BLUP, we estimated breeding values, I might choose a dedicated boar from that breed, or that line. Now I can use the dedicated boar based on an index that includes molecular markers. So we've really moved up the chain, if you like. We've gone from lines to individuals to now individuals chosen both on their breeding values and their genetic markers. Okay, well that's kind of where we're at, but I want to tell you where we're headed now and talk about where we're headed for the next couple of years. So this is a cartoon of DNA unraveling from a chromosome. And what we really want to know is, how do we sequence that? In other words, how do we tear apart the information on the chromosome, marker by marker by marker, or base pair by base pair, and understand what that means? And that's called sequencing. And that's the unraveling of DNA or genes to understand their function. And you hear a lot about sequencing. That's what's revolutionizing human medicine. You can go to the store now. Some drugstores are selling over-the-counter tests to see if you have some genetic predisposition for certain diseases. That comes from all the work that was done by sequencing to find those associations. So what can sequencing do? Well, if you look on your left in the screen, what you'll see is DNA sequence, all the different base pairs. What we really want to understand is why some of that allows those pigs to be small, and in other cases, some of the pigs to be large. And that's sequencing in its simplest form is going to allow us to do that. Now, sequencing also tells us all those different markers, all those different SNPs. And when we started out, and a good example here, I'll do this on the screen, in 1994 we essentially had two genetic markers, halothane and ESR. And then as we went over the years, sure, we got a few markers, and then we were talking maybe 1,000 markers, but most of these were under some type of private control or still under investigation. Well, sequencing, when we could sequence all of the all of the chromosomes of pigs, we could then get all the DNA information on 35,000 genes. And in doing that, we then actually created the scientists created a tool that would have 60,000 markers. So we went from a few markers to having a tool now for investigation, and that tool is like having 60,000 markers. Now you say, wow. Well, think about what's happened in other industries. Everybody remember what their cell phone looked like 10 years ago? It was like carrying around a shoebox. It was about that big. Now you have in your pocket a cell phone that has email, phone, video, God knows what else. Okay? Well, we've gone in the genetics business from a few markers in 1994 to now having a device, what's called a chip, that has 60,000 markers on it. Now, it's not ready for prime time. I want to be clear about that. It's ready for research. The companies are using it. Some of the companies are using it to try and understand their lines and so on. But this 60,000 SNPs, I can now genotype animals for a cost of about $200. ESR, the first genetic test, cost about $10 in the laboratory when it first started. They were selling halothane, I think, for $30. Okay? You could now do 60,000 tests for about $200 an animal. And with that, we're going to eventually get at things like trait associations, parentage, traceability, all kinds of things, and also something called genomic selection. So instead of this old-fashioned pedigree, which I'm sure most of you have seen a pedigree like this, we're going to do away with these kinds of pedigrees. And we're going to have pedigrees that look like this, where it may be important to the breeder to know the name of the animal, but to the geneticist, we want to know that string of interesting genetic code and how that's going to differ. And from that, we're going to then know how to predict where we're headed. Now, I'm not saying that this is available now, but 5 years from now, some of these tools are going to be in the hands of breeders. And it's going to be used for something that's called genomic selection. And Egbert had a great talk today about using breeding values to predict survivability. And the only thing that clearly is if you like, the roadblock to that is somebody's got to measure all those phenotypes. And that at some point is going to cost money. So what I'm going to propose now is a way to drop measuring those phenotypes, at least after the first generation. And it's called genomic selection, and let me walk you through it. So in the first generation, I'm going to collect phenotypes. I'm going to weigh all those pigs that he talked about. And I'm going to take as many measurements as I possibly can, and I'm going to genotype every one of those little piglets for at least 50,000 SNPs. And then I'm going to use a model to predict which of those genetic mutations, or SNPs, are associated with survivability. And there won't be one or two, there'll probably be 1,000 or 2,000. And from that, the next generation, I'm going to produce a lot more pigs, and this time I'm only going to genotype them. I'm going to tell those grad students of yours to go home. They don't have to measure those pigs again. If they're like my graduate students, they'd say, great, we don't want to do that. We don't want to sit there weighing pigs. And I'm going to use just the genotypes to predict the value. So instead of using phenotypes to predict breeding values, I'm going to use genotypes. And that's called genomic selection. And I'm going to keep doing that over generations. And I'm going to lose a little accuracy over time. And so maybe every 5 or every 10 years I'm going to have to remeasure those associations. But the point I want to make is the future will be in just taking a DNA sample, looking at the genetic differences, not in the 1s and 2s and 3 genes or 10 genes or 100 genes, but in the thousands of genes, and then use that to predict phenotypes. Now, not everybody's going to do that, but at the nucleus level, at the top of the breeding pyramid, whether it be seed stock producers or breeding companies, that's where we're headed. We're going to look at those 60,000 SNP markers, and we're going to perform what's called genome-wide associations. We're going to understand how those individual markers, as maybe these different colors on the chromosomes point out, their associations with different traits. Now, I often get asked, what's this worth? And this is my big guesstimate slide in the talk. People ask me, well, if I could find several markers for reproduction or thousands of markers for disease resistance, what would it be worth to the industry? And so I've put up some values here, and they are just best guesses. But I'm guessing for traits like growth, not a lot, because we can select for growth using standard measurements. Leanness, the same way. But disease resistance, if I can find several markers associated with PRRS resistance, that's going to have real value. If I can find markers associated with longevity, real value. Markers with reproduction, again, real value. And that's where we're headed. And whether you believe this number, double this number, or triple this number, it doesn't really matter. The point I want to make is we're headed towards using the genomic information not on a few genes, but on lots of genes. And this is going to produce, I think, some interesting products out there. And we always hear about vertical industries. We already know that you can look at the Tysons or some of the other companies that own pigs, bring the pigs to the slaughter plant, and then have ways to market those. It's vertical. They make more money because a loss at one place can be offset by a profit somewhere else. Well, I'm predicting that we're going to have vertical genotypes, or what I call designer genotypes, in pigs. So we're going to work with corn breeders. They're going to produce a specific type of corn that when fed to pigs is going to produce a product that is going to have real value. Now, some of you out there say, oh, that's never going to happen. Corn's a commodity, pork's a commodity, not going to happen. But it already does. If I go to Europe, I buy certain types of ham. Well, those are Iberian pigs that have been fed only acorns, and that system produces a ham that's being sold at about 10 times the value of a regular ham. So that's actually a certain genotype fed to a certain genotype to produce a certain product. And we're going to head that way in the U.S. for sure. Another place that's probably got more effect is what I call designer health plans. I know that takes on a bad name because everybody thinks about Obamacare, but in the pig business, we should be able to design genotypes that are what I call vaccine-ready pigs. We should be able to, in cases in which some vaccines work rather variably across populations, we should be able to determine which genotypes they work better on, select those animals so that when vaccinated, those animals will produce more immunity and higher resistance to disease. So this is another possibility that's coming down the pipe. Well, let me just kind of summarize what we've talked about. We've talked about ways to increase production. And the production increases are easily in the ballpark of 20 to 40%. Now, if you're a breeder and you're just getting started, you want to start testing for 3, 4, 5, 6 genes, with the idea that once you've incorporated those into your breeding plans, when the research is done with those 60,000 SNPs, you'll be prepared to move on to the next stage. We're talking about the eventual use of designer genetics to produce specialized crop and animal products. This is going to improve animal welfare. We're already starting to talk about selecting genotypes for certain, if we go away from the crate, we go to pen farrowing, we're going to need an animal that's going to be different. Well, let's start collecting the genetic information and look for genes that are associated with behavior. It's going to involve improved environmental conditions, because once we understand the genetics, the requirement of the pig, we can improve its environment. And I didn't get a chance to talk about cloned or transgenic animals. I don't think those are going to enter. I want to be clear about those. I'm not suggesting that's going to enter into the food source, but they might be. The pig might be a great animal to produce proteins and organs for human medicine. So that's something that might also be possible from advanced genetics. So actually, for the young people in the audience, and for the older ones too, I think there's an enormously bright future through genetics for improved food production and contributions to humans. So I want to thank everybody. I often get asked about this slide. Here's a bunch of pigs with barcodes on them. When we get done understanding once we get done sequencing pigs and understanding where we're heading, we'll eventually have sequencing down to just maybe $1,000 an animal. That's not going to happen to every animal in the population. But for the elite sires, we'll probably sequence them. And that sequencing will allow us to predict, just like a barcode does when you run a can through the supermarket and it picks up the barcode and tells you what the price is. We'll have that from a genetic standpoint in the future. So